Hey everyone, welcome back. And I just want to say, hopefully you had a good weekend. And we're going to start part 2 of protein synthesis so you guys can feel confident going into the next lesson, which we'll get to in a couple of days. All right, but quick review. Remember, in our last lesson, we talked about DNA turning into messenger RNA, because messenger RNA is responsible for carrying the message on how to make proteins to the ribosome. And so once it's in the ribosome, the protein can be made, and we learn how to find the different amino acids that form a specific protein. All right, so I'm going to go back and share with you the drawing from last week. Let me see here. All right. This video will be a little bit shorter than the last one, so don't worry. All right, so here's the paper we drew last time. Just a recap for you guys. We have our cell with a nucleus, our DNA turned into messenger RNA, carried the message to the ribosome. So this was our example of our first 3 codons. Remember, the word codon is every 3 letters. And when they were changed into RNA, we used our little decoding chart to find the name. So UCG, the name of our first amino acid, if you remember, was serine. So if you have that paper, UCG coded for serine. GUC coded for valine. So hopefully you guys were able to finish this entire paper yesterday. All right. The Nearpod had some questions. I think it wanted you to find AUA. Well, if I look at my special chart, AUA, I'm going to start here. AUA, so isoleucine should have been our next word. All right, isoleucine. GAU, GAU, looks like this one is aspartic acid. I'm going to abbreviate. And the last one, UGG. So I have UGG. So this one is tryptophan. So I'm going to abbreviate this one as well and just write TRYP. So if you got those, then you are on the right track. All right, a couple of things I just want to add to this lesson is we have to learn 2 new vocab words. All right, so what I'm going to do is get a new piece of paper, but you can feel free to write on your own piece of paper. I just ran out of room. All right, first term is called transcription. Transcription is just a fancy word, and you do need to remember this, but this is what it's called when DNA makes RNA. Okay, so transcription, DNA makes RNA, this occurs in the nucleus, right? If it involves DNA, it has to be the nucleus, because remember, DNA is too big, it can't leave the nucleus. All right, so transcription, that's the word. Now, the second word is very similar. It's called translation. Translation is when the RNA makes the proteins. And where are proteins made? You guys remember? It occurs in the ribosome. All right, proteins are made in the ribosome. So we have two words, transcription, translation. Transcription happens first, okay? Because DNA has to turn into RNA before RNA can make the proteins in the ribosome. So transcription is first, translation second. Well, what if you see these words on a test and you don't remember which one is which? The easy way I tell my students is look at the word itself. The first 5 letters are the same, T-R-A-N-S. The next letter is C for that word. The next letter is L for that word. If you ask yourself which word comes first in the alphabet, C does. C comes before L. So transcription has to come before translation. Okay, so just some new terms to teach you guys. Transcription happens in the nucleus when DNA makes messenger RNA. Translation happens in the ribosome, so that's when RNA makes the proteins. All right, one last thing I want to show you, and it's not the biggest thing that we have to know, but it is kind of important. Here's my cell, there's my nucleus, there's my ribosome. Okay, just you don't have to redraw this, but I thought I would show you. We already learned about messenger RNA. Let me choose a different color. Okay, there are 2 other types of RNA. One is called ribosomal RNA. The other one is called transfer RNA. So mRNA, you guys should know, carries the message, right? Carries the message to the ribosome on what proteins make. Our RNA, ribosomal RNA, is found in the ribosome. We don't have to know much about it, but it is in the ribosome. Okay. When we go back to messenger RNA, it carries the message. Remember, every 3 letters equals 1 codon, or essentially 1 amino acid. Okay, if you remember that term from last week. So on here, real quick, we have 1, 2, 3, 4, 5 codons, and we have 1, 2, 3, 4, 5 amino acids. So 1 codon is a code for 1 amino acid. Transfer RNA, this guy's kind of unique. And let me show you what I mean. Let's look back at our original sequence here. AGC and CAG. Okay, so I'm just going to write them right here. That's my DNA, just so we don't get confused. When it turned into messenger RNA, it was then UCG and GUC, okay? And really, I'm just taking these first 2, and I'm not changing them. So I'm using them again as an example. All right, so this messenger RNA, remember, goes to the ribosome. It's carrying the message. So I'm going to write it again here. UCG, GUC. All right, so these are my codons. This is what the code is for my specific amino acid. All right, this is where transfer RNA comes into place. Transfer RNA, I like to think of him as a little helicopter. He is actually the one carrying the amino acid. So here's my amino acid. All right, these are my codons. Let's not forget. Here's another codon. And the tRNA carries what we call the anticodon. Dun, 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 anticodon. All right, so if we were to decode this or find the letters, we are simply taking the 3 letters and using the same base pair rules in RNA to find the letters. So U would turn into A. C and G, remember, never change. So G would be my second letter. And again, G doesn't change, so it would be C. Now, a lot of people get confused and think that this is the code for the amino acid. This is not the code. Yes, my transfer RNA is carrying the amino acid, but he has to find the correct landing spot, like a helicopter. So this anticodon flies around till it finds the correct code or codon. He lands, he drops off the amino acid, and then the same thing happens with the next one. So if I have another tRNA carrying my amino acid, the anticodon for GUC would be C. U turns to A, C turns to G. All right, and so this one anticodon pairs to that codon, drops off the amino acid. Okay, so if we were to name these amino acids, again, remember our chart, we've already found them, so we don't have to do it again. The first one, UCG, codes for serine. So this would be serine. GUC codes for valine. All right, and as these all get dropped off, the transfer RNAs, they're dropping off all of these amino acids. And when they are complete, you have your protein. Okay, so pretty cool. All right, the transfer RNA is important when you're doing the worksheet, which I'm gonna show you briefly. But you also have to make sure that you definitely know transcription occurs in the nucleus, and that's when DNA makes RNA. Translation occurs in the ribosome. That's when RNA makes the proteins, okay? And we'll review this some more. All right, look at the paper that you guys have. Looks like this, okay? On the front is really just making sure you understand the structure of DNA. All right, so the phosphate group, they want us to color brown. So if you remember, this was our phosphate group. The 5-carbon sugar, they want you to color orange. Okay, this is my sugar. And the base, the nitrogenous base, they say purple. I just have a purple highlighter here, so we'll do that. Okay, so that's one nucleotide. Remember the word nucleotide. And then here we have a bunch of nucleotides connected, which makes up DNA. So the sugar phosphate backbone is going to remain brown and orange. So all of my circles are brown on both sides. And my sugars will all stay orange. Okay, I'm going quickly. You guys can do it neater. Now it gives me adenine, thymine, cytosine, guanine. Well, remember the base pair rules. Apples in a tree, cars in a garage. So adenine, thymine are going to be red and green. C and G are going to be blue and yellow. Well, Miss Conquest, which one is which? Does it matter? It does not matter as long as you stay consistent. All right, so if I have red and green, if adenine is red, thymine's green, let's just say I want the first two to be red and green. And then this one would be red. So my red, this would be adenine, this is my thymine. Notice how these are curved in the middle, these are pointy. So these cannot be A and T. They're different. So I would make these my G's and C's. All right, but here again we have curved. So my T and this would be A. Now on this side, it's still curved, but they switch it up on you. So this would be my T, this. Okay, this should be simple. You're just picking which one you want. If you wanted these to start with C and G, that's totally okay too. Just stay consistent as you work your way down. All right, that's the easy part. This is where it gets tricky. Let me get one that I haven't written on yet. All right, on the inside, it tells you what to do. Basically, we are going to change DNA into our complementary strand. So recall A, T, C, G. So they did the first one for us. A, T, T goes to A, G goes to C. The next three would be like that. So you want to fill this out going down. The next one is your mRNA. So here we have apples under a tree, cars in a garage. So C still goes to G. A cannot be T anymore. Remember, apples under, it's U. T still turns to A. So this is my codon. This is what's going to give me the name of my amino acid. All right, but they also ask for the anticodon. Okay, so if I'm going across, G turns to C, U turns to A turns to U. This is where people get tricked up. Your amino acid, the name of it, comes from the codon, the mRNA. Okay, so as you go down, every 3 letters, remember, is a code or a codon for the amino acid. So GUA is our first one. GUA, so that means this one would be valine. All right, so there's one example. You're going to do the next 3 on your own. On the flip side, it's the same thing, except now it's going horizontally. So DNA, remember AT, D, G. Apples in a tree, cars in the garage. So C turns to G, A, T, G, C. Now we're taking this and turning it into messenger RNA. G turns to C, T turns to A, C turns to G. The only time you have to be careful here is when you have like a T that turns to A. Remember, A cannot be a T in RNA. It has to be a U. So you should have no T's in this row, none at all. All right, now they also ask for the transfer or the anticodon. Okay, now it looks like this would be the code for my amino acid because it's attached. Don't get confused. This is the anticodon. So the name of the amino acid comes from messenger RNA. All right, so every 3 of these is going to give me the name of my amino acid. So the first one we can do together, CAG. CAG, we have what's called glutamine. So I would write that here, GLUTAMINE. All right, so then you'll finish the next 4. Then we have 8 questions for you guys to answer, okay? I'm not gonna do one with you because I want you to make sure you understand this. If you have questions, ask myself, Mr. DeWeese, or maybe a neighboring student who can help you. All right, so really the main thing we learned new today was transcription and translation. Hopefully you've written this down. And we talked about the transfer RNA, transferring the amino acids to the ribosome. And it's what drops off the ribosome or the amino acids to build the protein. but we only uncode messenger RNA, right? So we are uncoding mRNA only. All right, I feel like hopefully that makes sense. Let me see. One last thing. All right, there we go. So hopefully that makes sense. Your job today is to complete that worksheet. Make sure you have the DNA sequence with amino acids completed, and then you'll put those in the brown box. All right, hope you guys have a good day. Let me know if you need any help.